Okay, everyone should be seeing my slide. And if not, let me know. And in general, if you have any questions, just unmute yourselves and interrupt me. This is really a brief introduction to the world of modeling, at least as we see it here at CCAM. And I'm really interested in having an engagement with you. If you have any questions, you definitely should ask them, because hopefully everyone will be on the same page and will sort of understand the basic concepts after this brief 45 minutes. Many of you probably have already looked at the YouTube video. There's a YouTube video that's listed on this slide, but it's also listed on the schedule. So you can click on that and get more details about what I'll be saying. And I'd rather not repeat everything that's in that YouTube video. So if there is some discussion, that's very, very welcome. So anyway, let's get started. We're going to start with reaction kinetics. And let's see. This is just, something that you probably remember from freshman chemistry. We're just talking about a transformation of a molecule A to a new molecule B. It's a unimolecular reaction. It only goes in one direction. B cannot go back to A, at least in this simple model. So this is a model, and the molecule A has to overcome an energy barrier to get to B. That's why it doesn't happen instantaneously. There has to be enough thermal energy to kick A into B. And depending on how high that barrier is, that reaction rate will go slower or faster. So let's say that for this particular transformation, A going to B, there's a 10% chance for A to jump the barrier within 1 second. And that's a rate constant. That means that the rate constant is 0.1 per second. So if you had 10 A molecules at time 0, how many B molecules would there be at 1 second? Well, you can kind of assume that there'll be around 1, maybe 2, maybe 0. The fact that we can't say for sure means that this is a stochastic problem. This is a stochastic model. If we have a very small number of molecules, we can't really say for sure whether any one particular molecule is going to be able to jump that barrier. We'll gain enough thermal energy during 1 second to jump that barrier. But if we have a lot of them, we can say that about 10% of them will jump the barrier in 1 second. Okay, that's because the rate constant is 0.1 per second, or 10% chance for A to jump within 1 second. Again, if anyone is having a problem with that, we don't have a huge group here. You should be feel free to ask a question. So basically, I'm giving you the definition here, or giving you an understanding of what this concept of stochasticity is. That fundamentally, we're dealing with individual molecules. And when you really consider the behavior of a small number of individual molecules, the behavior is probabilistic or stochastic. And we can make a good guess at how the system will behave. But every time we look at the system, trying to experiment, for example, with a small number of molecules, the answer may come out a little different. So that's probability, stochasticity, etc. If we have a large number of molecules, let's say trillions or millions, then we can have a pretty good idea of what that system will look like with very little error after that 1 second. So if we express the system in terms of concentrations instead of individual particles, then that becomes a deterministic system, and we can solve this problem if it has a rate constant of 0.1 per second. You can turn it into a differential equation. The change in the concentration of A with respect to time would be minus 0.1 per second times the concentration of A. So if you solve that little differential equation, the solution is A is equal to the initial concentration of A times an exponential decay with a decay constant of 0.1 per second. So that's the deterministic version. And again, this should be kind of familiar to everyone from your undergraduate chemistry courses. But if it's not, please interrupt. Okay, so that's sort of the definition now of deterministic. It's deterministic, really not fundamentally deterministic, but if you really have a large number of molecules and you're comfortable expressing the system in terms of concentrations instead of individual particles, then it can be solved with differential equations. So just to show you what that looks like in a little bit more detail, on the left is a solution for those 10 molecules that we originally discussed. A goes down sort of erratically. And as I said, if we did that experiment again, it would come out somewhat differently. There might be not as long a gap between 7 seconds and 16 seconds. You see that long gap right where the A label is. But the next time we did it, there might be a short gap for those kinds of transitions. So it's going to change every time. The deterministic solution, which you see on the right, is, of course, a solution to an equation which is always going to be the same. So that's why we call it deterministic. If we, instead of using 10 molecules, we use 1,000 molecules, there's a little bit of noise in the trajectories, but they begin to look very much like the deterministic solution. The more molecules you have, the smoother the stochastic solution will be, and the more it will converge to the deterministic solution. Now, depending on the model, that isn't necessarily always going to be true, but for simple models, that's generally true. Okay. The kinetics of complicated networks can be simulated either stochastically or deterministically, whichever is appropriate. As Michael showed you in his introductory slide, you have to decide that based on how many molecules you've got in your system. And then you can use numerical methods for both stochastic and deterministic models using either Kopassi or V-cell. Both will give you basically the same capabilities, the same solutions for these kinds of problems. So here we're showing a very complex network. This is a screenshot from Virtual Cell, and this is a screenshot of a deterministic solution in Virtual Cell where we're looking at all these various species that are represented in the model. Okay. So the nice thing about Virtual Cell or Kopassi are that they can solve these very much more complicated problems, which you can't do by hand like you can solve for a simple exponential decay, they can do this for you automatically. Another important concept is the concept of steady state. When all the rates become zero, you reach a steady state. So here's a simpler reaction, more complicated than the exponential decay, but a reaction where the molecule A and the molecule B come together to form a complex called C. And then C can undergo another reaction where C becomes D, okay? So this is a little bit more complicated, but it's still fairly easy to grasp. And if you start off the system with both A and B, and they combine and start producing C and D, there's going to be a series of curves that you can look at for the change in the concentration of A, the change in the concentration of B, and C and D, etc. A and B will start going down if you start with them, and C and D will start coming up. Now, let's say that some of these reactions are reversible. So if as C builds up, it can dissociate back to A and B. So there's going to be a reverse rate constant. And in this rate expression for reaction 1, I show that the forward rate is K1 times the concentration of A times the concentration of B, simple mass action rate expression. But there's also a reverse rate where C can go back to A and B, okay? And likewise for the transformation of C to D. The forward rate for this reaction is K2, a different rate constant, times the concentration of C minus, this was a little mistake here. This should have a K minus 2, the reverse reaction times the concentration of D, okay? So when all of these reactions level off, you've reached equilibrium, you've reached a steady state. And at that point, all of the rates approach zero. And it's very often important to know what the steady state of a system is. And you can calculate that in a number of ways. It's really just algebra if you consider that there's also conservation of mass. The overall number of molecules of each type has to be constant, has to be not constant, but it has to be conserved. So if you start off with 1,000 A molecules and 1,000 B molecules, you can't produce 2,000 C molecules. You can only, at the most, produce 1,000. So those are called conservation of mass relationships, and those are all algebraic equations that you can solve for. And Copassi does that for you automatically. Another way to do it is you can start a kinetic simulation and let it run until all the species reach a constant value. And that's your steady state, okay?
Excuse me, sir.
Yes.
Sir, for the particular example that you mentioned, for that one, we are trying to calculate the steady state. For that, we have to find out the forward reaction, the rate of forward reaction of the C, and then the rate of forward reaction of C to D.
Right. You would need to know all those rate constants. And
right.
in general,
Got
when
it.
you build a model, you need to know the rate constants. You need to at least make a good guess at the rate constants. So those are the concentrations, and the rate constants have to be the inputs to your model. Does that answer your question?
Yes, sir. I got it. Thank you.
Okay, good. So as I said, you can also start a simulation and let it run till all the species reach a constant value. And an important thing to know, to keep in mind when you do when you build your models, very often when you build a model, you are interested in the response of the system to some sort of stimulus. The stimulus could be the addition of a ligand to your system that binds to a receptor. So what happens after time point 0 where you've added the ligand? That could be a stimulus, or it could be an electrical stimulus if you have an electrophysiological model. Whatever the stimulus might be, that's what you're really interested in, right? So it's very important that before you add your stimulus, that the system is at steady state. Because if you have a system like this that you see in this little reaction diagram, and let's say you add a stimulus where you start destroying molecule D. You add you can even do this by sort of, let's say D is photochemically active, and you flash it with light, and D becomes destroyed. How does the rest of the system change after you start photoactivating or photo bleaching or photo damaging molecule D? Well, if you haven't reached the steady state yet, then you can't really answer that question, because the system hasn't relaxed, and it could be changing due to these other reactions, not just to the destruction of D. Okay? So before you apply any kind of stimulus to your system, you have to make sure that it reaches a steady state. And that can be simply done, as I said, by letting the system run for long enough so that it reaches a constant value for all of the molecules. And then you can just use that as that steady state as the initial concentrations for the virtual experiment where you apply the stimulation. Everyone follow that? Any questions on that issue? It's an important concept, because people very often make a mistake when they're modeling by applying the stimulus at time 0, even when the system hasn't reached steady state. Let's say they start A and B at certain concentrations, and they don't have any C and D, and they also apply the stimulus then. Well, everything's going to change because A and B haven't had a chance to react yet. Okay, so I'm emphasizing that point because, as I said, it's often something that people have problems with.
I have a question. In capacity and V cell, does it automatically solve at steady state when you're like changing when you're running simulations and you change like a specific thing? Is it already assuming that your system's at steady state, or is that something you need to like check for?
You need to check for it. You need to be sure that I mean, you can go to steady state. As I said, Kapasi has some algebraic routines that allow you to calculate the steady state before you add your stimulus, or you can just run the simulation out to steady state. You can do that either in Kapasi or VCell, and let it just reach steady state. You can do that in either system. But once you're there, that's when you want to apply your stimulus. So yeah, I mean, you can calculate the steady state using Kopassi or V-cell, but it won't automatically do that. It won't automatically, I mean, you have to know what your initial concentrations are. Your system would be constrained by having certain concentrations, say, of A and B in this system, because you know that you're never going to get by conservation of mass, you're never going to get more C than you have A or B, right? You're never going to get more D than you have A or B. And all of that is automatically dealt with within both software systems. Did you want to add something, Pedro?
No, I was just going to say we can talk more about this when I describe Kopassi. In Kopassi, there are some automatic ways of getting steady state. So for some things this work, but we will talk more about that in detail later this morning.
Okay.
Thank you.
Could I also ask a question, Les?
Sure.
Would it be okay if you just ran simulations for, say, 1,000,000, 10,000,000 or 100,000,000 seconds or something first? If, say, you have some reactions that mean that you're never really going to reach a steady state, do you feel like that's an acceptable way to equilibrate a system?
Well, I mean, in general, I would say no. If you really have to run it out to 1,000,000 seconds to reach something that still isn't a steady state, then you really need to think more deeply about what the system is. But if you want to apply a stimulus, I mean, if you know what you're doing, and you know that you're never going to be at steady state, and you still want to see what the effect of a stimulus is, as long as you're conscious of what you're doing, then that's fine. You can,
Okay.
Just have to, when you write your paper, you need to explain that to people.
Yeah. Okay. Okay. Thanks, Les.
Sir, I have a very basic question.
I see a
May
question.
I ask?
Sorry, Mayfaz, what?
Sir, I have a very basic question. May I ask?
Yeah.
Sir, when I'm practicing simulations, I mostly think that when I get a straight line, for example, I get a simulation that gives a value, I run a simulation at 1 second and it gives a value of 65, and it is constant throughout. Whenever I increase the simulation time, it remains constant. Should I consider it as a steady state or not?
Well, all of the variables in your system need to be constant, and if that's the case, then you could consider that a steady state, yes. Not just one variable, all the variables need to be constant. Okay, how do you pronounce your name? Selvik or?
Yes, correct, Selvik. So I have a question. The time the system reaches the steady state is dependent on the initial concentration one chooses and the rates, but it will always end in the same steady state? Or can a steady state be different for different initial concentration and rates?
Yes, the steady state can be different for different concentrations and rates. So you have to determine it for each set of concentrations and rates.
Okay.
And even for the same set of rates, you may have multiple stable states depending on the initial conditions. Again, we will maybe be able to talk about this later on when we have a session on steady state.
Okay, thanks.
This is an introduction, so we're just giving you the basic ideas, but you should be sure that you understand these basic ideas. That we're going to get more complicated later. Okay. Any other questions? Okay. Okay, now let's get to diffusion. Molecules move from place to place, even when they're not reacting, even when there aren't any reactions. So this is just some animations showing individual molecules. So this animation starts when you see this red line here, and the red line goes away. That's a barrier. And the question is, how long does it take for molecules to get across the barrier? Now, if the molecule, there's just single molecule, it's bouncing around with a certain rate that's given by its diffusion coefficient. And in this case, it never gets to the other side of the barrier. But when you have many molecules and some of them started off near the barrier, then there certainly will be several molecules that get to the other side. And furthermore, when you have large numbers, where again, we're dealing with concentrations now instead of trying to count individual particles, there could be there's a smooth transition where molecules get depleted from the left side of the barrier and start accumulating on the right side of the barrier. Okay. And that accumulation or that change can be described by Fick's first law, which simply says that molecules move down a concentration gradient on average. And so the deterministic solution to this, I mean, the probabilistic solution, the stochastic solution, is given by following each molecule individually using probabilities. You know what the average movement of the molecule is based on its diffusion coefficient, so you can calculate a probability that a molecule will cross a barrier. But if you have large numbers, you're in better shape because then you can solve it using differential equations. And the differential equation for that corresponds to Fick's first law is that the flux across that barrier is just given by a diffusion coefficient, a constant, the proportionality constant, times the gradient of the concentration, the derivative of the concentration with respect to the distance. And so this is sort of, I hope this is sort of intuitive, that molecules move down a concentration gradient. But what's not, maybe you haven't been so familiar with, is the idea that this is also fundamentally a stochastic process that you can describe deterministically when you have a large number of molecules. Okay. Now, there's also Fick's second law. Fick's second law is, the way I like to describe it is that stuff accumulates if that stuff moves into a region faster than it moves out. That's all it is. That's Fick's second law. But if you want to describe it in terms of a differential equation, it has a little bit more of a complicated expression. This is Fick's second law in 3 dimensions. It says the change in the concentration with respect to time is given by that same diffusion coefficient for that particular molecule times the second derivative or the second partial derivative of the concentration with respect to each dimension. This is now a 3D version of the equation. Okay, so again, all that Fick's second law says is that if molecules are moving into a region faster than they're moving out, then they'll accumulate in that region. And likewise, if they're moving out of a region faster than they move in, then they'll start going down in that region. And you can kind of see that if you look at the barrier, stuff is moving into the region right to the right of the to the left of to the right of the barrier, and it's moving out of the region to the left of the barrier because of the second derivative of the concentration with respect to X in this situation. Okay. So I go into that a lot more in the YouTube video, but really that's all there is to it. And it's, again, something that you're all sort of, I think, experientially familiar with. Now, when you have reactions coupled to diffusion, then you simply have to add the kinetics to fix second law. And so this is the kind of equation that you would write down, and the kind of equation that virtual cell will solve for you. So virtual cell solves spatial problems as well as non-spatial problems. And so for this kind of a problem where you do have to worry about diffusion, you would use virtual cell. And it can solve these problems either deterministically or stochastically. You can follow individual particles in a space, like in, the volume of a cell, or on the surface of a cell, on a cell membrane. And you can also solve for these kinds of problems deterministically using concentrations, and even hybrid versions where some particles that are present at very low concentration, single particle numbers, can be followed stochastically, and other molecules that are present at very large numbers can be followed deterministically in terms of concentrations. So all of that can be done in virtual cell. And the question is, when do you need to use virtual cell? When do you need to use partial differential equations to solve a reaction diffusion problem? This kind of equation is called a reaction diffusion equation. And there are two conditions that you need to consider before you run a full reaction diffusion problem. And you don't want to use reaction diffusion. You don't want to use partial differential equations or spatial stochastic modeling unless you really need to, because the simulations take a lot longer. Okay. So you really, it's important to think about what you're doing and not use a method when it's not necessary. So in order to decide when you need to use it, there are two considerations. First of all, diffusion has to be slow on the time scale of the reactions. If diffusion is very fast on the time scale of the reactions, then the molecules involved will spread out within the entire volume before a reaction has a chance to happen. And if that's the case, you might as well consider the concentration to be uniform within the volume, right? Everybody follow that? So if the diffusion is very fast on the time scale of reactions, you don't need to use partial differential equations. You don't need to solve for space. You do need to know how big that volume is, which I'm sure you'll learn more about later. But you don't necessarily need to solve for the diffusion explicitly. The other thing that seems kind of obvious that you need to consider before you solve for the full reaction diffusion system like this is that the distribution of the species is not uniform within the cell. So at the starting point, before you apply your stimulus, say, the molecules that are involved in the model in the system that you're interested in are not uniformly distributed. If they're completely uniformly distributed within the volume of the cell, for example, then there's never going to be a diffusion gradient, right? So you need to consider that. But very often, cell biological processes start off by stimulating molecules in the membrane. And then there's a transmission of that information to molecules in the volume, right? And if that's the case, then the system is not uniform. If a membrane is involved in the model, then you can be pretty sure that the system is not uniform. So so if there is a stimulation at a membrane and that triggers reactions in the cytosol, then that might be necessary to solve for diffusion and to do this full reaction diffusion system. But both of these considerations have to be considered before you decide to do these more computationally intensive simulations that are done with virtual cell and using reaction diffusion equations. Okay. So I'm going to give you a couple of examples, and then I'm actually, I think I have enough time, unless there are more questions, to actually give you a demonstration of this within virtual cell. So let's consider that you have this sort of rectangular cell, and that there's a phosphorylation that occurs on one side of the cell that releases a second messenger that starts diffusing away from the site of phosphorylation. Okay. So in the first instant, clearly there's going to be a little bit of a diffusion gradient. And we've satisfied that one condition that we had, that the system is not uniform. We're starting off the stimulation at the membrane. In fact, in this case, only on one part of the membrane. And we're going to ask what happens. So let's see what happens in terms of the second condition. Is diffusion fast or slow? So if you have a fast reaction and slow diffusion, then as the system evolves, you can maintain a concentration gradient, because the molecules are being produced here faster than they can diffuse to the other side of the cell. If you have a slow reaction and fast diffusion, then even though there are molecules being produced here, they're diffusing so fast that they're filling up the cell uniformly. And you can consider this system without worrying about diffusion. So this kind of system can be dealt with just with ordinary differential equations or non-spatial stochastic simulations. And this situation does require full spatial type simulations. Okay, any questions on that? Okay, when is diffusion slow on the time scale of reactions? So just a little rule of thumb, that the average distance that a molecule moves is given by is proportional to the square root of time. It's given by the square root of 6 times the diffusion coefficient times time. So that gives you an idea of what the time scale of diffusion is. And the time scale of the rates, you can think about that as 1 over those expressions r sub I. 1 over r sub I is sort of a time scale of a rate. So you can sometimes estimate that. Estimating this might be somewhat difficult. But if you actually run a compartmental model, an ordinary differential equation model, you can get an idea of what 1 over you can get an idea of what the time scale of the rates is just from running that. So another sort of piece of advice as you develop your models is, even if you suspect that a model might require explicit consideration of diffusion, might require a full reaction diffusion treatment, you should still start off by getting an idea of how the model works by doing ordinary differential equations, by not worrying about diffusion. Okay. So we have about 4 minutes before I stop, so I want to give you a little demonstration in VCell. Everybody can see this? So I have this little model here in VCell. Hopefully your screens are high enough resolution to see. Maybe I'll make this a little bigger. So it's a simple model where a receptor produces a transcription factor, and the transcription factor starts off at the membrane. So this gray thing here is the plasma membrane. The transcription factor starts off at the membrane. It diffuses to the nuclear membrane, and then it gets transported into the nucleus. Okay. And the transport into the nucleus is one way. So there's it's a pump that's pumping it in. And we have a simple model here where this is a little bigger, where we've mapped this system to just a simple sphere. So here's a surface view. So we've got a sphere with a green nucleus in the middle of the sphere, okay? And let's look at the simulations. So I have 2 simulations here, one with D equals the diffusion coefficient equal to 10. And we know that this system satisfies the first consideration, that is, it's a non-uniform system. The molecules are starting at the membrane. So let's go down to the middle of the sphere. Okay, this is about the middle. And this is 0 time. Let's look at the transcription factor. At 0 time, it's 0 everywhere. If we go down to 10 seconds, there's a very nice gradient. And I can do a chimograph. This is giving you a little preview of what virtual cell is like. So I can do a chimograph where you can see that along that line that I drew, this is the distance along the line in the x-axis, and down the y-axis is time. So you can see that the system reaches a steady state and that there is a significant gradient that persists, because as the molecule diffuses to the nuclear membrane, it's being pumped in. So there's always a depletion of the molecule at the nuclear membrane. So this system clearly requires full reaction diffusion if you're going to ever see that gradient. Let's look at this one where I've used a much higher diffusion coefficient of 500. By the way, a diffusion coefficient of 10 in the first system is sort of what the diffusion coefficient of a protein might be. Diffusion coefficient of 500 is the diffusion coefficient of a small molecule, like a second messenger, like calcium or something like that. So let's go down, do the same thing. We're going to go down to the middle. And let me go down to the transcription factor. And you can see that the gradient is much smaller. Okay, very little gradient. If I do the chimograph, this will be the last thing I do. You can see that it does reach a steady state very quickly, but that the gradient, if I put my cursor over here, you can see that there's very little change in going from the plasma membrane to the nuclear membrane within the cytosol. There's a tiny gradient, but it's not very significant. And this kind of system probably could have been solved using just ordinary differential equations with very little error. Okay, I think I'm just about done. And to learn more about virtual cell, I'll turn it over